Hello, my name is Casey Adam. I'm a postdoc with Antoine Jerusalem, and we study the cellular mechanisms of neuromodulation. Now, ultrasound neuromodulation is a very promising new treatment where we use what is essentially a mechanical and heat stimulus to alter the electrical activities of the brain. However, what ultrasound is doing at the cellular level still remains a little bit unclear, and hopefully clarifying what's happening there can help us improve ultrasound neuromodulation in the future. Now, ultrasound is by no means the only neuromodulation agent that we don't fully understand. For example, we've been using anesthetics, which are chemical neuromodulation agents, for centuries, and we still don't have a clear picture of how they work. And while there is a whole diverse array of neuromodulation agents and they all seem very different, the hypotheses we've proposed to explain how they operate are surprisingly similar. And these hypotheses often involve changes in the intermolecular interactions or the subcellular structures of various components. So these are things like changes in ion channels or changes in the cell membrane or cytoskeleton and a variety of other subcellular components. Now, because intermolecular interactions give rise to the mechanical properties of the cell, what we've essentially been hypothesizing is that neuromodulation agents alter the mechanical properties of neurons and that this alteration somehow changes the electrical activity of the cell. And at first, this might seem a little bit counterintuitive. However, we've known since action potentials were discovered that they're not solely electrical like we often treat them. They are, in fact, a coupled phenomenon. So not only do the electrical properties of the cell change during an action potential, but the mechanical properties, chemical properties, pH, and heat also change as the action potential propagates. And these changes are all coupled and never exist independently of one another. So if we start to study neurons from the lens of this coupled phenomenon, and specifically from mechanoelectric coupling, we might be able to gain some better insight into neuromodulation. So with that goal in mind, my lab uses a custom experimental setup shown here, where we use a nanoindenter to measure the mechanical properties of individual cells. Specifically, we're interested in the storage modulus, loss modulus, and loss tangent of the neurons because these quantities relate to energy storage and dissipation by the cell. And in particular, we're interested in the loss tangent, the ratio of energy dissipated to energy stored, because the loss tangent affects force propagation and attenuation. So the loss tangent might actually be playing a role in attenuating the action potential as it goes through the cell. We can also use the nanoindenter to apply a mechanical stimulus to an individual cell. Similarly, we use a patch clamp to measure the electrical properties of cells or impose an electrical stimulus to the cell. We use an inverted microscope and techniques like calcium imaging to film cell firing dynamics, and we've enclosed everything in a gas-tight box so we can expose cells to various chemical neuromodulation agents. So with this setup, we can employ the various tools at our disposal according to experimental needs. And today I'm going to be telling you about 3 experiments that we've performed on this setup. In the first experiment, we wanted to see whether a mechanical stimulus at ultrasound frequencies altered the electrical properties of cells. So in this experiment, we performed a patch clamp in current clamp mode to measure the membrane voltage of a cell, and then we used the nanoindenter to apply a mechanical stimulus to that cell. We found that when the stimulus was applied, cells initially stopped firing, and then when they resumed firing, they exhibited faster action potential dynamics and were hyperpolarized. So once we've demonstrated the mechanical to electrical side of things, the obvious follow-up is whether an electrical stimulus would alter the mechanical properties of cells. So in the next experiment, we essentially reversed what we did in the first experiment. So now we performed patch clamp to impose a voltage over the cell, and then we used the nanoindenter to measure cell mechanics. And we found that when you impose a positive voltage on the cell, both the storage modulus and loss modulus of the neuron decrease. However, this decrease occurs in a similar manner, so the ratio of the two, the loss tangent, remains constant. So after these 2 experiments, we've shown and we've quantified mechanoelectric coupling in neurons. So the next question is whether neuromodulation alters both the mechanical and electrical properties of cells. And to test this hypothesis, we decided to see whether a chemical neuromodulation agent, isoflurane, affected cell mechanics and firing. So to expose cells to isoflurane, we use a setup that is reminiscent of what you would find in the clinic. So in this setup, you load liquid isoflurane in a syringe. When you turn on the syringe pump, that liquid is pumped into a vaporizer here, where the liquid isoflurane vaporizes into a gas. The gas then flows into our airtight box containing the sample, where it interacts with the cells and flows out into a waste scavenging unit. So essentially, by turning the syringe pump on and off, we can start and stop exposing the cells to isoflurane. And that is what we do in our experiments. So first, we measure cell mechanics and firing before they're exposed to isoflurane. We then turn on the syringe pump and we measure cell mechanics and firing 15, 30, and 45 minutes during isoflurane exposure. We then turn off the syringe pump, and as the cells recover, we measure their mechanics and firing 30 and 45 minutes into this recovery period. Now, in this setup, we also employ two controls. The first control is to make sure that we can distinguish the effects of isoflurane. And so in this control, we simply fill the syringe with room air, and we have the same flow setup that we have with isoflurane. In the second control, we want to control for the effects of gas flow. So in this control, we fill the syringe with room air, but we do not establish any kind of gas flow through our experimental system. And we found that the gas flow does indeed affect neuron firing. I don't have enough time to get into these changes, but if you're interested, please feel free to ask me questions at the conference. But suffice it to say that if we want to see the effects of the isoflurane, we need to focus on the comparison between the flowing systems, so the flowing isoflurane or the flowing air, the black line and the red line here. And we found that upon initial exposure to isoflurane, both the fraction of cells that fire as well as the number of times cells fire decreases. However, with longer exposure to isoflurane, both quantities increase again to similar to the air control. So what we've shown is that isoflurane alters cell firing. It does not dampen cell firing. And these measurements actually match what we've seen in EEG studies, where firing patterns change, but they aren't completely dampened when someone is exposed to isoflurane. So after measuring these changes in neuron firing, we wanted to see if isoflurane altered cell mechanics as well. And so again, we did the same setup where now we measured neuron mechanics before, during, and after exposure to isoflurane. And as you can see in the first row, isoflurane exposure increased the neuronal storage modulus. In the middle row, isoflurane also increased the neuronal loss modulus. However, the increase was to a lesser extent and with a 15-minute delay compared to that of the storage modulus. Now, because the loss modulus increased less than the storage modulus, the neuronal loss tangent actually decreased. So now that we've seen changes in cell firing and mechanics, our next question was whether these changes were related to one another. So we did some regression analysis, and we found that the neuronal loss tangent correlated with the number of times that neurons fired. So, in conclusion, we have quantified mechanoelectric coupling in neurons, and we've shown that neuromodulation alters both cell firing and mechanics. Also, because our measurements are performed on cells as a whole, we've demonstrated that it's really the cell as a whole that is altered by neuromodulation. So when we focus on a particular ion channel or a membrane, that's only a small part of the picture, and we really need to see how that piece fits into the cell as a whole if we want to truly understand neuromodulation. We've also demonstrated that there's a correlation between the neuronal loss tangent and the number of times that cells fire. And then finally, we've shown that it's important to consider the coupled properties of cells when we're trying to understand how neuromodulation works. So with that, I would like to thank my supervisors, Antoine and Kathy. I would also like to thank Celine and Miran, the other scientists on this project. I'd like to thank Ari for his help with our isoflurane setup and James for machining the airtight box. Finally, thank you all for listening, and I look forward to any questions.